The classical method in which membrane proteins are extracted for their study is by simple solubilization in detergent. While this provides a simple and universal method, detergents have been shown to alter the native structure, function, and activity of these sensitive proteins. Here we present the Peptidisc as a solution for stabilizing membrane proteins in detergent-free solution. The Peptidisc system spontaneously adapts to the size, shape, and topology of myriad membrane proteins. This stands in contrast to other membrane mimetics that often require tedious optimization.
Few A, an E. coli outer membrane protein, will be used as a model target protein to be reconstituted in this protocol. Outer membrane vesicles will be isolated, followed by the addition of detergent to solubilize the membrane proteins. The Peptidisc peptide is added to the solubilization, and detergent is diluted away, spontaneously forming Peptidisc particles. These stabilized soluble particles are now amenable for numerous downstream applications. This protocol will examine biolayer interferometry as an application of the Peptidisc technology.
This protocol will focus on the PeptiQuick reconstitution method, a useful technique that facilitates the simultaneous purification and reconstitution of membrane proteins.
The PeptiQuick method of on-beads reconstitution is performed in a standard nickel-NDA iMac gravity column pre-equilibrated in detergent. The solubilized membrane is loaded onto the resin and non-target proteins are washed away. Peptidisc peptide is added to the column and the detergent is diluted rapidly. The hydrophobic side of the Peptidisc peptide associates with the exposed hydrophobic regions. The hydrophilic side of the peptide keeps the protein